Okay, so we talked about DNA replication. Now we're going to talk about transcription, which is going from DNA to RNA. Okay, RNA is actually most of the nucleic acid that's found in the cell. It's transmitting the information from the DNA into protein form. It's like the middleman. We talked about the differences to DNA. First of all, the base, it's uracil instead of thymine in RNA. The big thing is the sugar, right? The deoxyribose is missing that oxygen. RNA has an oxygen on that on carbon 2 of the sugar. Okay. The reason why there's this RNA middleman is to keep the DNA safe, right? The DNA like lives in a cage in the nucleus. The RNA is the copy of that same information that then gets transported outside the cell into what's called the cytosol, or the rest of the cell, where it's going to be transcribed into proteins, right? But the whole idea is you want this middleman so that you can keep your DNA locked safely in a cage and then spit that part out of the cell. You don't want to expose the DNA to the rest of the cell where something could go wrong. Okay, types of RNA. The one that most of the RNA is actually found in what's called ribosomal RNA in the ribosomes in the cell. The ribosomes are the actual machines that are making proteins. Okay, so most of the RNA in a cell is actually in these ribosomes because these ribosomes are everywhere. All right. There's a ton of them. They have a lot of RNA in them. They're actually enzymes, but they're enzymes that contain both amino acids and RNA and nucleic acids. Okay. So most of the RNA in the cell is actually locked in these machines, these enzyme type machines that are actually doing the protein synthesis. Another one is the transfer RNA, which is the component that builds the amino acids. So ribosomes rather, use tRNAs to link amino acids together. All right, and we'll talk about a lot more about that in the next lecture when we talk about transcription. This is all about translation, going from DNA to RNA. Okay. The last kind of RNA is what's called messenger RNA, and that's which is only 5%, but it's arguably very important because that messenger RNA is what contains the information of the DNA, right? So when we talk about going from DNA to RNA, we're specifically talking about making mRNAs, which are going to tell the ribosome what proteins to make. Okay, so the mRNA contains that information that's read by the ribosomes. And that's the same information that was in the DNA. Again, we just don't want the DNA out into the rest of the cell. So we first go through this middleman, this mRNA middleman, spit that out into the cell, into the cytosol, and that's what's read by the ribosome. The whole goal of transcription is to copy the same information that's in that DNA. The DNA contains what information should be in the RNA. All right, and specifically, again, that mRNA. Once the mRNA is made, it's going to be a copy of what that DNA is, or at least a complementary copy of what that DNA is, and then it's going to be kicked out of the nucleus into the cytosol. All right, and we'll talk a lot more about the role of tRNAs next time. Let's focus first on the making of those mRNAs, right? So this translation process going from mRNA to the protein, that's what the next lecture is. We're going to focus on this transcription process here, which is going from DNA in the nucleus, transcribing it into mRNA. A lot of times the mRNA that's made has little segments that are clipped out called process. It's called a processing step. We're not going to focus too much on that. We're just going to worry about how the DNA is transcribed into RNA, specifically again, mRNA. Okay. So first of all, the DNA has what's called a section. primer section that sort of indicates this is where a gene lives. So start here, right? Most of your DNA is actually not red. It's kind of just there for structural reasons. You could see a lot of our evolutionary history in part of our DNA. There's only a small subset of your DNA that actually gets red, right? And by being red, I mean transcribed into RNA. Every single segment that gets read is called a gene. And there's a little section before it that says, hey, there's a gene here, start reading, okay? Proteins will bind to that primer section, and that's what lets mRNA or RNA polymerase, similar to DNA polymerase, but RNA polymerase, which makes RNA from a DNA template, lets that bind. The mRNA that's being synthesized, it knows what segment should come next because of that complementary base pairing. Okay. The only difference is that now we're going to replace uracil with thymine, right? We don't use T in our RNA, we use U. Once that mRNA is formed, it gets kicked out of the nucleus into the cytoplasm. Cytoplasm is like the rest of the cell. Okay. So again, there's some sort of a region here that would say, hey, there's a gene here, start binding. The RNA polymerase would recognize that section and bind to it, and then it's going to start reading that DNA template and creating RNA by adding RNA linkages that, again, you could see they're going to match the DNA template strand, right? So my template DNA is ATGCT. My new RNA or mRNA that's being added is going to be complementary to that, again, having U instead of T being added. All right, but that's how the cell knows what mRNA unit to add next. Same way that DNA polymerase did it, RNA polymerase does the same thing. It looks for complementary base pairing. Okay. Regulating the mRNA synthesis. So, just take a step back. Eventually this mRNA is going to code for a protein, right? And when we talked about enzymes, we talked about the one guaranteed way to increase the reaction rate of an enzyme catalyzed reaction is to increase the concentration of enzyme. We talked about how enzymes themselves are regulated through sort of feedback inhibitors and all that good stuff. The genes themselves are also regulated. So if the body wants more enzyme that breaks down sugar, it will make more mRNA that corresponds to that enzyme. Okay. So the gene itself is regulated, and it's done by what are called transcription factors, those things that recognize, hey, there's a gene here, and bind and start the whole process. Okay. So here's a sort of an mRNA strand. Again, this promoter region, this is the region that says, hey, there's a gene here. If the cell wants to make more of a particular protein, it'll create more transcription factors that recognize that promoter region and allow RNA polymerase to bind. Okay, so RNA polymerase is just sort of, it binds when it recognizes one of these transcription factors. The transcription factor is a protein itself, and it recognized a particular promoter region on a gene, on a strand of DNA. So that promoter region lives right before the gene, so that RNA polymerase can bind and start reading. That gene will be transcribed into mRNA, which will then be kicked out of the cell and translated into a protein. And again, that translation step is the subject of next lecture. Okay, so promoter region is just a region on the DNA. It's just a particular sequence that's recognized. So AATTG, let's say that that's a promoter region. There's a protein floating around called a transcription factor that is meant to bind to any time it sees AATTG. Does so, and that encourages RNA polymerase to come on and then read the gene. Okay, so So just, to summarize, transcription factor binds to the promoter region of the DNA strand. That transcription factor allows RNA polymerase to bind to the DNA. and RNA polymerase starts recognizing, synthesizing this mRNA strand based off the gene, the complementary base pairing of the gene, which is the DNA strand. Eventually, it'll hit this sequence that it recognizes as a termination sequence and fall off. So there's going to be another stretch that RNA polymerase can't hold on to, I guess is one way to think about it. It recognizes it as being the endpoint, and so it stops and it'll fall off. So you always get only your particular gene is going to be copied. Nothing. You want to start and stop at an exact point. Okay, so if I gave you the sequence of a strand of DNA, and I wanted to know what the complementary strand for mRNA would be, here you have multiple options, but we don't really even need them, right? So I'm going to, again, so it's going to be complementary, meaning we're going to go in the other direction. Cytosine and guanine, T is complementary to A, but then because we're making RNA instead of DNA, A would be complementary to U. C. So that would be this option here.